1.
Education is the most powerful weapon which we can use to change the world. Good morning to one and all present here. I warmly welcome you all to the international webinar on Introduction to PCR and its application in diagnostics, organized by the Department of Plant Biology and Plant Biotechnology. Now, I warmly welcome our principal, the man with the Midas touch, Dr. M.G. Raghunathan sir, to deliver the welcome address. Sir, please.
Good morning, one and all present here. Most esteemed resource person of the day, Dr. A. Khaleefathullah
Excuse me, sir. Your voice is not audible, sir.
Gopi, unmute.
Okay, sir. Nice.
Can you hear me? Okay. I'm sorry, it was on mute. Somebody unmuted. Now I have unmuted. Good morning, one and all present here. Esteemed resource person of the day, Dr. A. Khaleefathullah Sheriff. And Dr. Gopi, head of the Department of Plant Biology and Biotechnology, Dr. Poonam Sethi, organizing secretary of this program and the host of this program, and galaxy of professors and invitees and student friends who are all attending this webinar through YouTube. A hearty welcome to you, one and all present here, and a special welcome to Dr. Khaleefathullah Sheriff from Oman, who has really accepted our invitation in spite of this busy schedule. Sir, a hearty welcome to you, sir, on behalf of Guru College. And then the topic given by the Department of Plant Biology and Biotechnology is Introduction to PCR and its applications in diagnostics. Even the layman would have understood the importance of PCR now because of this COVID. It got popularized. The topic is need of the hour, how this PCR can be. Even in our college, we have a PCR kit, but we have not used it. Hope this webinar will explain the usage of PCR for all the people so that it can be utilized in a proper way. With this, I wish and congratulate the Department of Plant Biology and Biotechnology for choosing a wonderful topic of the international webinar. My best wishes to Dr. Gopi and Dr. Poonam and the team of galaxy of other faculty members of the Plant Biology and Biotechnology. Once again, a hearty welcome to the resource person and also all the participants. Thank you. Thanks for the opportunity. Thank you.
Thank you, sir. Now, I call upon Dr. M. Gopi, sir, head of the Department of Plant Biology and Plant Biotechnology, to give few words about the webinar. Sir, please.
Most revered General Secretary and Correspondent, most respected our beloved Principal, sir, benevolent head of the department, professors, and my dear participants. A pleasant good morning to everyone. I extend a very sincere warm welcome to all of you. I am extremely honored and feel privileged to have given the opportunity to address everyone on this special occasion. Firstly, I would like to extend gratitude to our dynamic and vibrant principal who has granted us permission to conduct this webinar. On behalf of our Guru Nanak College management, principal, and our own department and student, it is my great pleasure to welcome you all for this wonderful international webinar on Introduction to PCR and its applications in diagnostics by Dr. A. K. Sheriff. Today, we will highlight the importance of PCR in modern world. Recent developments in molecular methods have revolutionized the detection and characterization of microorganisms in a broad range of medical diagnostic fields, including virology, mycology, parasitology, microbiology, and dentistry. Among these methods, polymerase chain reaction has generated great benefits and allowed scientific advancements. PCR is an excellent technique for the rapid detection of pathogens, including those difficult to culture. Along this conventional innovation and is playing an ever-increasing role in clinical diagnostics and research laboratories. Due to its capacity to generate both qualitative and quantitative results, real-time PCR is considered a fast and accurate platform. In this virtual webinar, I recognize some familiar names of participants from different sectors, states, and countries. I am sure this webinar will be need of the hour and fruitful for every one of you during this pandemic situation. I sincerely hope you will enjoy this webinar. Thank you. Have a nice day.
Thank you, sir. Next, I call upon Dr. Poonam Sethi, ma'am, to give introduction of the resource person. Ma'am, please.
Thank you. I would like to start with a quote. A good teacher can inspire, hope, ignite the imagination, and install a love for learning. Brad Henry. Now, I would like to start. The influence of a good teacher can never be erased. When I was doing my PhD project in King's Institute, Gindi, Sir A.K. Sharif sir was there who helped me a lot. Now, regarding his about him, he has over 50 publications and nearly 500 citations. He is actually called the man of influenza. Human influenza virus, any doubt you have in King's Institute, you can go to Sharif sir and clear your doubt. And not only influenza, he's got expertise in dengue, chikungunya, Japanese encephalitis, Coxsackie, and polio virus. Okay, regarding his qualification, he's got a postdoc from Pittsburgh, USA, in the year 2012 in Thomas E. Transplantation Institute. He's worked as a research scientist 1 medical under ICMR project monitoring of human influenza in India from 2005, 2006, 2007 to 2010 and 2012 to 2015. Currently, he is in Oman working as a specialist A microbiologist. Now, this it doesn't end here. He's got, to his credit, which is worth to be mentioned, he's worked as a scientist in WHO collaboration project and CDC Atlanta from the year 2002 to 2005. He's done surveillance studies for influenza virus among the Chennai population. He's also worked as a research person in the project on Coxsackie group B virus RT nested polymerase chain reaction, which is funded by the prestigious Lady Tata Memorial Trust Mumbai from 2002 to 2005. And is also the awardee of Young Investigator from the year on the year 2011. And he's also won a science fellowship by the Avian Society, Bristol. Now, this is regarding his educational qualification. He has handled poliovirus, influenza virus, Coxsackie virus, arbovirus, hepatitis virus, then HSV virus, measles virus, dengue virus in most of the WHO and CDC accredited laboratories. He's got a technical skill in immunology, animal handling, stem cell culture, molecular biology, virology, and in microbiology. And we have to also mention that he has worked with the software like Al-Shifa, PLIFA, which is Polio Laboratory Information for Action, and MLIFA, Measles Laboratory Information for Action. He has also authorized a book, Influenza Diagnosis Manual, in the year 2006. That is Avian Influenza Diagnosis Manual 2006. Not only this, now regarding about him, he himself from the horse's mouth, he wants to nurture the best of his skills, both as an individual and a team. So with this, you can find out that he really is a very good leader, and he sees to that not only him, even his team comes up. And he also, according to him, he wants to set goals and serve the mankind irrespective of the clan through the influential or inevitable research of science. So he wants to really bring up mankind or make mankind disease free. So thank you, sir, for this accepting our invitation. Thank you. Welcome you, sir. Vinosha, thank you.
Thank you, ma'am. Without further delay, now I call upon today's person, A.K. Sheriff, specialist in medical microbiology from Muscat, Sultanate of Oman, to give lecture about into PCR and its application in diagnostics. Please.
Good morning, everybody. Thanks for the very elaborate introduction, Dr. Poonam. It's really a privilege for me to be giving a webinar in a esteemed institution, a college which really enjoys or very good thoughts in me for a pretty long time, right from my days when I was studying. So we'll start with the webinar for today, Introduction to PCR and its application in diagnostics. The contents of today's talk will be the nucleic acid extraction, the principles involved in it and the types, the different types of PCR and its principles, and the diagnostic application of the PCR. The objectives of my talk today will be to understand the basics and principles of the methods of nucleic acid extractions and the various systems and the instrumentations which are used in it. Learn about the basics and different types of PCRs which are used for diagnostic and research, and know about the applications of PCR and appreciate it. So first, what is a molecular test? A test that checks for certain genes, proteins, or other molecules in a biological specimen is called as a molecular test. Molecular tests can also check for certain changes in a gene or chromosome that may cause or affect the chance of developing a specific disease or disorder. So in this, we can know that the molecular test can be used to check presence or absence of a certain gene. For example, if you're looking for a COVID-19 sample, whether the patient is positive for it or negative for it, it can detect. At the same time, it can also check for certain changes in the genes, like if there is any variant, like now a lot of variants like Delta plus are coming up in COVID. The molecular test can also help in detecting the variants and also to check any mutations that result in any abnormality or disease. I have illustrated the different molecular tests based on the complexity. The simplest one is the single-plex PCR, in which we just amplify one target gene. And the slightly complex one is a multiplex PCR, in which we detect for more than one target. Then we nowadays the syndromic approach is very much prevalent in the diagnostic system. In the syndromic approach, what happens is we do an in the syndromic approach, what happens is we do a panel of tests for different pathogens. For example, when you do a respiratory panel, it will detect for the viruses causing respiratory disease, as well as the bacteria and fungi all together. Similar way for blood culture organisms and for gastroenteritis panel like that. And then comes the detection of viral loads. In this, we will be able to quantitate the amount of the pathogen that is present in the biological sample. This is mainly useful to know how the treatment is working. It helps mainly for the prognosis. And still more complex tests are the genotyping of the viruses. And the most complex one for now is the sequencing, which helps us to detect the new variants. Basically, molecular testing can be divided into three main parts. The first part is the nucleic acid extraction. Then it is followed with by the amplification of the extracted nucleic acids. And the third part is the detection and resulting. Nucleic acid extraction. To do a molecular test, particularly a PCR or sequencing, we need to have a template, which is the nucleic acid. So there are different methods by which we can extract the nucleic acids. The basic method or the primitive method or the first initial method, which was used for extraction of nucleic acid were the solvent-based methods. Though this is primitive, this formed the basis or the base for all the modern technologies to develop. Then the advancement came, which involved the use of a solid phase, like a spin column based extraction assays, which is slightly advanced than the solvent based one. And these can be used in the lab with low workload. Then the third type is the magnetic beads, which is now being widely used in all the diagnostic laboratories. This is very rapid, and this gives us the purest form of the nucleic acid. And this has a flexibility that it can be used in both high and low throughput labs. So first I will discuss a few things about the solvent-based extraction, nucleic acid extraction method. The first step you have to do is to disrupt the cell walls by grinding. And once the cell wall is disrupted, we mix the disrupted material in the lysis buffer or the extraction buffer, and this has to be thoroughly shaken, either using a vortex or with the help of a homogenizer. This helps to break down all the cell membranes or dissolve the cell membranes. Then the crude lysate, which is obtained after homogenization, contains the nucleic acids and other cell constituents. This has to be mixed with an equal volume of organic solvent, either a phenol or chloroform or a phenyl chloroform mixture. Then it is centrifuged. After centrifugation, we get three phases. The top aqueous phase contains water-soluble molecules, including the nucleic acids. The proteins and lipids become trapped in the organic phase, and they're thus separated away. And the plant debris become trapped in the interphase. So now we have to collect the aqueous phase. This step is very important because we should not contaminate the aqueous phase with any of the other phases, or else it will lead to erroneous results and the low purity of the extracted nucleic acid. Once we separate the aqueous phase, we have to add an equal volume of alcohol with salt to precipitate it, and then centrifuge. After centrifugation, we'll find that there is a pellet of nucleic acid, and then it has to be washed a couple of times with 70% ethanol to remove the residual salts and other contaminants. Then it can be dried and dissolved with, and the pellet can be dissolved with water or tea to obtain the nucleic acid. If you don't want to use phenyl chloroform, you can directly add alcohol to the cell debris, centrifuge, pellet, and then take it as the other steps. This is a bit cumbersome, but it may take at least it may require at least one overnight incubation to get the maximum yield of the nucleic acid. The next one is the spin column based extraction. Here the first step, again, it is the same. We should have a lysate combined with alcohol, and it is placed into a spin column and then centrifuged. This results in the removal of proteins and divalent cations, and then it is washed a number of times to remove the impurities, and pure DNA is finally eluted from the membrane into the sterile water or tea. So this is how we have the lysate with the alcohol. Then we add it into the spin column. This has a silica gel on it, and then it's centrifuged. Once the centrifugation is done, the nucleic acid gets trapped in the silica gel, and the other debris and other things will be coming out in the elute. Then this can be washed a couple of times with different buffers of different compositions and concentrations to remove as much as impurities we can. Then the column is spin column is dried, and we add TE or distilled water or elution buffer, and then centrifuge and collect the elevate. This has the quality of the nucleic acid obtained by spin column is much better than what we get by using the solvent-based extraction methods. Here, one thing you have to keep in mind is we keep throwing the elevate from the first, second, third, and fourth steps. But finally, in the final step, what we elute contains the extract of nucleic acids. So we had to be a bit more concentrating on the last step so that we don't throw the elevate and make a mess of it. So generally, this is a mistake which people do when they do the extraction by using spin column methods. So the last step, you have to focus a bit more and then collect the elevate and store it. The instrumentation required for spin column based extraction is very simple. Just with high speed centrifuge, you can able to do the extraction. But if you want to go for an automation, you have options as an instrument called IQAC from QIAGEN. It does an automated extraction from using the spin columns. Then the final thing in the extraction, the latest method which is being widely used is a magnetic bead based extraction method. Here, the sample is mixed with the lysis buffer and magnetic beads are added to it. The positively charged magnetic beads selectively bind to the DNA under optimized conditions. And then the same way as we do in the spin column, there are two washers to remove the unwanted contaminants. And finally, the nucleic acid is eluted into the elution buffer. This gives us the purest form of the nucleic acid, maybe DNA or RNA for extraction. This is one example of the instrumentation which is used for the magnetic bead extraction. This is a machine from a company called Life River. This can extract 12, 24, or 36 samples in 20 minutes. But you have to do a bit of pre-processing, which may take around 20 minutes. So in 40, 45 minutes of time, you will be able to extract around 36 samples. And this is the plate which they provide us. And this is a preloaded plate. Preloaded plate in the sense the plate has everything loaded in it. It has the magnetic beads, the lysis buffer, the washing buffers, and the elution buffer also. So what you have to do is you just have to add the sample with the carrier molecule and the protease. Then keep it into the machine, and the machine will process it in 20 minutes and give you the purest form of nucleic acid. This is a robust machine called Kingfisher. It's the best machine in the sense that it gives the most purest form of nucleic acid. It is also magnetic based one, and in 20 minutes this can extract 96 samples. So this is ideal for the labs which are doing a number of samples, and it's very, very important during the times like the pandemics, when we have to work on a lot of samples in a shorter period of time. But the main issue with this machine is that everything you had to do it by yourself. You see, you can see these deep well plates. You have to add everything by yourself. One plate completely for the sample and the lysate and the beads. The next plate, the wash one. The next plate, the wash two, and then the elution buffer. So you have to add everything and then keep it in the machine, and the machine will process it very fast. The pre-processing time will be around 1 hour, and then 1 hour and 20 minutes for the machine. So in 1 hour 20 minutes, you'll be able to extract purest form of nucleic acid from 96 samples. This is another machine which is a bit simpler to work with, but it's very slow. It takes a long time. It takes a long time. It takes a long time in the sense it takes around 4 hours for sorry for it. It takes 4 hours for the completion of the 96 samples. So now we have finished the first part of the talk, which was mainly focusing on the different extraction methods and what are the differences between the methods and which method to be used where, like that. And now we move to the next step, which is the amplification of the extracted nucleic acid. How do we amplify the nucleic acid? We amplify the nucleic acid by the help of PCR. So the topics that I'll be covering in this are: what is a polymerase chain reaction, the history and prehistory of PCR, how the PCR works, and the different types of PCR and its applications. What is a polymerase chain reaction? Polymerase chain reaction is a means of selectively amplifying a particular segment of DNA. The segment may represent a small part of a large and complex mixture of DNAs. It can be thought of as a molecular photocopier. For example, when we collect a nasal swab from a patient suspected of COVID and do the extraction of the nucleic acid, the extract will not only contain the COVID-19 genome if the patient is positive, but apart from that, the patient's own cells, the nucleic acid from the patient's own cells, or the nucleic acid from the common cells which are present in the patient, or the nucleic acid from any other pathogen which the patient has been infected, uh, everything will be present. So the extract will be a complex mixture of nucleic acids. So from that, we'll be amplifying our target gene or our target region, which is what done by the polymerase chain reaction or PCR. PCR is a very powerful tool. It can amplify an amount of DNA visible by gel electrophoresis in approximately 2 hours. They call it like finding a needle in a haystack. If you're going to search for a needle in a haystack, you see it, it's not an easy just to find it if you're searching for that single needle. But what PCR will do is it will amplify the needle so many times that it will outgrow the haystack and you'll be able to find a needle easily. This is how we can explain what the PCR does in the best way. The template DNA need not be highly purified. You can just use a boiled bacterial colony, but this is a generalized statement. If you're looking for a very common gene, this is possible. But if you're looking for something very specific or very rare gene, you have to definitely have a purified form of the nucleic acid. The PCR product can be digested by use help of restriction enzymes, and it can be sequenced and cloned. PCR can amplify a single DNA molecule provided the molecule is pure and not degraded. PCR was invented by Caramellis in 1983. The first published account of it appeared in the year 1985, and he was awarded the Nobel Prize for Chemistry for this invention in the 1993. Did he really invent PCR? Yeah, he really invented it, but a long time before him, the basic principle of replicating a piece of DNA using primers was already described by Gopi and Khurana. But he couldn't progress further because of the lack of infrastructure for primer synthesis, and he couldn't find any pure polymerase. Mullis had the idea and had the infrastructure, and he really exploited it to invent the PCR. So what is in the reaction? So the PCR reaction, we have the main thing is the template, that is the nucleic acid we extract from the biological sample. Then we have the reaction buffer, which contains like trace ammonium ions, potassium ions, magnesium, etc. Then the nucleotides, which are nothing but the building blocks of the strands, which are amplified during the PCR. Primers are a short strand of DNA or RNA that serve as a starting point for DNA synthesis. And these primers are the ones which determine the specificity of the PCR reaction. Intact DNA polymerase, which is, we can call it as a mason of a building. Like it is the one which works in adding the nucleotides and synthesizing new strands. But nowadays, no need to worry about all these things because we have kits which just provide you with a buffer mix, an optimized buffer mix and an enzyme mix and water. You just have to add the extracted nucleic acid and the required quantity of enzyme and buffer mix and load it into the machine, and it amplifies. There are three main steps in PCR. They are the denaturation, annealing, and extension. What happens during denaturation is the machine heats the sample to 95 degrees so that the double-stranded DNA are separated into the single-stranded DNAs. And then annealing happens at 55 to 60 degrees, in which the primers, if they find a complementary region, they will bind to it. So the primer binding takes place at 55 to 60 degrees. And then extension, once the primers are bound to the target nucleic acid, then the tag starts to add the nucleotides, and it progresses continuously. And this, the time of the extension depends on the size of the product. If you're amplifying a smaller product, it will be around 30 seconds. If you are amplifying a larger project, maybe 1 minute or more than that one. So these three steps of denaturation, annealing, and extension are repeated at least 35 to 45 times. And how does a polymerase know to stop when it reaches the other primer? I will play a video at the end of the presentation when you can have a better idea of what all I have spoken now. How many copies? No target particles are made until the third cycle. So initially, there are some products which are not of the exact size which you are looking for. It will be slightly bigger or smaller. And then the accumulation is not strictly at the doubling phase. In the early phase, in the early phase of the PCR reaction, there will not be a doubling of the copies. And at the end of the reaction, there will be a major chunk of the required or the target particle or the product with some non-specific products also. There are three phases in the PCR. They are the exponential, linear, and the plateau phase. The exponential is a very important phase of the PCR reaction because it has an 100% efficiency and exact doubling of the products happens during this phase. And this is a phase in which all the reagents are in the optimal condition and we get the best products or the best results. Then the exponential phase is followed by the linear phase, which is of high variability. The reaction components are being consumed and the PCR products start to degrade during this phase. And the final is the plateau phase, which is an endpoint analysis. At this phase, the reaction would have stopped. And if it's left for a long time, the PCR products will start to degrade. So the optimal cycles are like around approximately 35. What happens if we increase the cycles to 45 or 50, thinking that we'll be getting more products and better results? No, that will not happen in a PCR, because already by 35th cycle, the plateau phase would have occurred. The plateau phase occurs because the reagents which are there are depleted and the products tend to re-anneal and the polymerase is damaged. You see the reagents in the PCR solution, the PCR mix, are being subjected to different temperature conditions with a short within a short period. They are denatured at 95, annealed at 55, and again extended at 72, and again going for a denaturation. So this different temperature changes really has an impact on the reagents, and it leads to the degradation of them. So it's not generally recommended to go for more than 35 cycles. These are some examples of the thermal cycler. So after we run the PCR, that is a conventional PCR, we have to run it on a gel to visualize the result. So the product which is amplified moves on the gel, and by using a molecular weight marker, we determine the size of the product which has been amplified. And if it is within our target range, then it means it is a positive reaction. If it is not within the target range and it's out of our target range, it means that some non-specific amplification has happened. And if you don't get anything, it means that the sample is negative. And one thing while doing the gel electrophoresis, you have to keep in mind is never add ethidium bromide to the gel, or avoid adding it even to the buffer. Better to run the gel, visualize it using the tracking dye, and then stain it with the ethidium bromide solution. Because as the ethidium bromide intercalates with the DNA, so it doesn't allow the proper movement of the amplified product in the gel. So the best way to have the proper molecular weight determination will be to first run the gel and then stain it with the ethidium bromide. So is PCR easy? Yes, now it's very easy because we just use the kit, load the reagents and put it in the machine, and after 1 hour or 1.5 hours, we get the results. But when it started, it was very difficult. The cycling was performed with 3 water baths, one for denaturation, annealing, and one extension like that. And thermal cyclers were introduced in the year 1986, but even at that time, they didn't have thermostable polymerases. Only after the discovery of Thermus aquaticus, and then the tag DNA polymerase in 1988, the PCR kits became easy to use. So what will be the optimal concentration of the different reagents in the PCR reaction mix? Template DNA or RNA ranging from 100 to 250 nanograms in 1x reaction buffer. DNTPs for 200 micromole concentration. And primers are ranging from 0.1 to 0.5, and DNA polymerase, usually it's tag 0.25 to 0.5 units. Then what is multiplex PCR? As I told you in the beginning, this is a slightly complicated compared to the singleplex PCR. So here, if you're going to detect, if you're suspecting a patient for influenza, he may be having influenza A or influenza B. And influenza A, what does he have? Influenza A H1 or H3 or H2. And the same time, if he is positive for influenza B, is it B Yamagata lineage or B Victoria lineage? There are different types of things in the influenza to detect, to know about the influenza virus. So if you're going to run each and every sample separately in different tubes, then we'll be ending up using 10 tubes for one reaction, for one sample, I mean. So instead of that, we can multiplex it. Multiplex it in the sense we can use the primers for the different genes in the same tube, and we can detect influenza A, pandemic or non-pandemic in one tube, influenza B, Victoria or Yamagata lineage in the other tube. So this is called as the multiplex PCR. And the products obtained will be of different molecular weight. And so based on the molecular weight, we can tell by visualizing on the gel whether it is influenza A or B or which subtype it belongs to. Can PCR amplify an RNA? Not directly, but the RNA has to be first copied or converted to cDNA by reverse transcription. And then it is being accomplished by an enzyme called a reverse transcriptase. And thus once the cDNA is ready, the tag can start its work and do the amplification of the cDNA. You can do it in 2 ways. First, you can amplify the cDNA separately and then use the cDNA as a template and run the PCR. Or else you can go in for one step in which the program is set in such a way that the first step will be the amplification of cDNA into amplification of cDNA, and then it will be followed by the PCR cycles. Real-time PCR. This is the revolution in PCR. The most important thing in this is that there is no post-processing required. What I mean here that no post-processing is required is no need to run the gel after finishing the PCR. Okay, if you don't run the gel, how will we read the results? We have one extra element in this and one extra reagent. It is called as probe. A probe is a fragment of DNA used to detect the presence of specific DNA fragment within the sample. The probe is smaller to a primer, and it is being labeled with the fluorescent dye. So this helps to, this emits the signal, or it fluoresces when there is a target molecule found in the sample, and based on the increase in fluorescence, we detect the presence or absence of the virus or bacteria or whatever we are looking for. So the different chemistries which are used in real-time PCR are Cybergreen, FRET, etc. And the main advantage of the real-time PCR is that it detects the amplicons in the exponential phase. Why this is important? Because this is the phase in which we have the 100% specific products, whereas in the endpoint PCR, it is in the plateau phase, and there will be a lot of other unwanted products also. But in the real-time PCR, it detects the product amplicons in the exponential phase. It quantitates the product and is able to detect the twofold change. It is very good in quantitation. As I told you, to detect the viral load or whatever it is. If you're doing it by conventional PCR and running the gel with ethidium bromide, it is not as sensitive as real-time PCR. If there is a twofold change in the concentration, if you have 1,000 and 2,000 copies, the ethidium bromide will not be able to tell you, whereas the probes which are used in the real-time PCR are sensitive enough to detect even a small change, that is a twofold change. This is mainly used in viral quantitation, viral detection, bacterial detection, and drug therapy efficacy, etc. Some details about the Cybergreen technique. This is the simplest form of the real-time PCR. Here we use a non-specific dye called Cybergreen. The Cybergreen attaches when there is a double-stranded DNA. So whenever it finds double-stranded DNA, it attaches to the strand and it fluoresces. But the problem here is it is not specific. Whatever double-stranded DNA it sees, it will bind and it will fluoresce. So we have to make sure the product to which the Cybergreen has bound is our target molecule or not. To know that, we have to do a melt curve analysis and check the melting point. And if it matches with that of our primers, then yes, that is the right product. And if the melting temperature is not the same, then it is not the product of our choice. It is some false amplification. Next is the next chemistry of in the real-time PCR is using the FRET FRET principle. The expansion of this is fluorescence resonance energy transfer. This uses two dyes, a reporter dye and a quencher dye. When these two dyes are in close proximity, the fluorescence emitted by the reporter dye is quenched by the quencher dye. But when they move apart, then the fluorescence will be then the quencher will not be able to quench the fluorescence of the reporter dye, and it will be visible for the optic system to measure. This principle and the 5' exonuclease activity of the Taq polymerase together has resulted in the making of a probe called Taqman probe. So the Taqman probe will have a small nucleotide strand which is complementary to the target sequence which we are looking for. And one end you will have the reporter dye, and the other end you will have the quencher dye. So once the probe is there, and if the sample has our target nucleic acid, the primer will bind to it and the probe will also bind to it once during the annealing. So after annealing of primer, the tag polymerase starts to add the nucleotides to the primer, and it extends the strand. As it extends the strand, it sees that there is something on its way, and it has an 5' exonuclease activity, and it starts to cleave the probe. Once the probe is cleaved, what happens is that the reporter dye is released, and as it moves away from the quencher dye, the fluorescence is emitted and it's captured by the optic systems, and it results in the positive reaction. So if there is if the sample is negative, there will be no template DNA for the primer and probes to bind. There will be no cleavage of the probe and no fluorescence. So if the probe is intact, you can see the fluorescence is very low, and the probe is cleaved, you can see that the fluorescence is high. There are other different types of techniques like molecular beacon, hybridization probes, eclipse probes, all are based on the same principle of FET. So CT value, what is the CT value? When you get any report, PCR report, you'll find either the copy number or CT value. CT value is always seen in the qualitative PCRs. So the cycle in which a significant increase in the reporter's signal above the threshold can be detected. That is the cycle in which the growth curve crosses the threshold. In simple words, the cycle in which the positive sample moves away or breaks out from the background fluorescence, it is called as the CT. In this, if you see, till the cycle 22, in the cycle number 22, you can see that there is a peaking of the fluorescence. So it means that this sample has a CT value of 22. This is data how a PCR run looks like. When you, this is a standard which we generally use for the quantitation purposes. The high concentration and followed by descending concentrations, low, and the lowest one, like that. Okay. One thing you have to note, the raw data will look like this. It is in the log settings. Then this is called as a threshold line. Once the experiment is finished, you have to visualize it in the log view, and then you just pull the threshold line up to the mark where there is no background fluorescence. And here, another thing I like to emphasize here is that if the CT value is low, it means that the concentration is high. Why is it so? The concentration of the pathogen is so high that it's being detected early. So if the CT value is 5, 10, or 15, it means that the patient has a high load of the pathogen. Whereas if the CT value is 30, 35, or 40, it means that the patient has a very low load of the pathogen, because it takes a long time for the machine to detect. So to summarize the differences between the traditional and conventional PCR, the traditional and the real-time PCR. The traditional PCR is measured at endpoint, that is the plateau phase, whereas the real-time PCR collects the data in the exponential or growth phase. It's low resolution, the size-based discrimination only in the traditional PCR. Ethidium bromide first chaining is not very quantitative, and it requires post-PCR processing, and detection is capable only to a 10-fold change. But in the real-time PCR, an increase in reported fluorescence signal is directly proportional to the number of amplicons generated. No post-PCR processing detection is required, and and detection capability is down to a 2-fold change. So this is very important, the 10-fold change and 2-fold change. So if we have 1,000 copies and then 10,000 copies, only then the fluorescence in the ethidium bromide will be able to show a difference. Whereas when you have even a two-fold change, like from 1000 to 2000, the real-time PCR will be able to show it. Some example of real-time PCR instruments. And I'd like to mention about some point of care instruments also. They are the Gene Expert, BD Max, and Panther. They are found in the market. And these are very easy to perform. You just had to load the sample and keep it in the instrument, and it will run and give you the results in 1 or 1.5 hours. But the problem with this is they are very expensive, and they cannot be used for high volume of samples. So wherever we don't have many samples, or if you have to make some urgent decisions, like if a patient comes, you have to admit him and you want to know whether he can be put into the COVID ward or into the normal ward, and you need an immediate answer. At those times you can use this, or it can be used in some remote places where you get only very few samples, and it's not worth to establish a full-fledged molecular biology lab. In those places you can use these systems. So the points to be kept in our mind are that always use a prototype virus. Only if you use a prototype virus, you will know whether the primers which you have used have worked or not. Then use the no template control or water extraction control with just distilled water. This will rule out whether there is any contamination in the reagents which you are using. And use a reaction control. Reaction control is nothing but you should have an housekeeping gene, because to ensure that the sample has been collected properly, and during the extraction steps, the sample has not been degraded. So all these controls are very important in a PCR reaction. You always use nuclease-free labware, use filter barrier tips to prevent cross-contamination, and use low retention tips for not wasting the reagents, and use other general good laboratory practices. The application of PCR, you all be knowing now that it is being used for the screening of diseases, particularly after the advent of COVID. So diagnosis and screening of diseases like HIV, hepatitis, testing for mutation, like for influenza, we have amantadine resistance. So we do a PCR to detect whether there is a mutation. That genetic profiling in forensic or legal and biodiversity applications. As I told you, now there is a trend of performing syndrome-based assays, because the symptoms which we see in different patients are overlapping with different pathogens. For example, in a respiratory disease, the symptoms caused by influenza or some other viruses will not be much different. So we should not waste our time by checking for influenza, and then if negative, then going for the other one, RSV or adeno, like that. So we have got a lot of panels which detect for around 20 to 25 pathogens at the same time. And there are many kits available for the detection of bacteria, fungi, and for detection of cancer and other issues. The pros and cons of the PCR are that the molecular tests are always quicker and have less turnaround times. The sensitivity is much improved, and accuracy is increased. They are becoming more user-friendly with automations, and built-in monitoring system ensure the quality of results. So it has many advantages, but at the same time, there are a lot of disadvantages also. They are expensive, but when you look into the cost of hospitalization and the life-saving time which we get by rapid detection of the pathogen, this expense can be compensated or justified. but it needs a dedicated expertise. It's not that everyone just can go and do a PCR, and it needs dedicated laboratory spaces. The laboratory space is the major issue we find everywhere. So the conventional PCR system needs three distinct areas to avoid contamination. So there has to be an universal flow, like the first area, the first term should be for massimic preparation, followed by the extraction, and then the third one should be for amplification analysis. So the flow should be unidirectional. So first you make the master mix and give the plate to the extraction room where the extracted nucleic acid will be added to it, and then it is being transferred to the next room, which has the PCR machines. And so the take-home points from this webinar should be like, traditional methods for extraction, though cumbersome, have formed the base for modern technologies. We need to have a thorough knowledge about them to understand the principles and develop our skills. The molecular methods are rapid, sensitive, and specific, but they are expensive also. Good laboratory practices have to be followed strictly to get optimal results. If not, it will lead to a lot of unwanted errors and contamination. PCR is a very powerful tool, but it needs to be handled with discipline, or else it will misbehave. So whenever you do an experiment, if you fail or if you don't get the desired results, please don't get disheartened or abandon the experiment. You just have to have one thing is that we learn more from our mistakes rather than by doing the things correctly from the first try. So never ever give up. Keep trying till we succeed. And I thank the organizers, the respected principal, and the HOD and the team behind this webinar for giving me an opportunity, and for all the participants.
Thank you so much, sir. We have few questions that are posted in the chat box.
Yes.
The questions will be popped up in the studio screen, sir. So will you be able to manage reading by yourself, or may I help you, sir?
Yeah, I'm seeing this like.
Yeah, thank you.
There is a question from Dr. E. Gayatri. How can I calculate the expected size for amplification PCR product in a PCR reaction? So this you have to run a gel, and the gel, agarose gel, and the concentration of the gel will vary according to the product size which you have amplified. If you have a smaller product, then you have to have a more concentrated agarose gel. But if you have a bigger product, you should have a lower concentration of agarose gel. And then you will be using the molecular weight markers. Here, you have to make sure that when you're in the selection of your molecular weight markers, that if you're going to look for a plasmid DNA, then you have to use a plasmid-based molecular weight markers. If you're looking in for just a double-stranded DNA, you have to use the markers for that one. So based on the molecular weight marker, you can compare your product's length and come to a conclusion. How can we detect live or dead pathogens using PCR? No, live or dead, we cannot differentiate whether the pathogen is alive or dead using PCR. PCR just detects the presence or absence of the nucleic acid in the sample. Again, the question, how to determine a plasmid copy number using qPCR? Yeah, it is based on the ethidium bromide fluorescence. You can detect it, but there will be softwares available. There are softwares available with the gel documentation system. By using that, you can do that one. How to analyze chip PCR data? Sorry, I don't have an idea about it.
So you have few questions even in the studio pop-up screen. Like the question is like, sir, in RT-PCR report received by COVID-19 positive patient, CT value of different genes, that is NORF 1A,
Okay.
yes,
Yeah, it's
etc. is
valid.
shown differently.
Yes.
What does it mean?
Okay. Now, for a diagnostic PCR, we use more than one target sequence because we have to make sure that we are not going to miss the miss the detection of the positive sample. So we use the three genes like N, ORF, S genes for COVID PCR, and these genes vary from the manufacturer of the kit. Each manufacturer have their own genes. So ORF, S and N gene. N is a nucleocapsid gene, open reading frame, and S is the spike protein gene. So the different values in this means that uh, how much amount of the particular gene is present in the patient. So generally we'll find that ORF is always high because it is found in more concentration in the sample, and then the S and N will be almost similar. For example, if you get an ORF reading of 35, then S and N will be around 37, 39, something like that. So, but once it is positive, it means it's positive. That's it. Okay. CT value prediction is made through CT scan. Why not through PCR now, sir? No, sorry, I can't get the question. Certain required tool sets. Okay, the question of Shweta Balakrishnan. The PCR you're talking about here is called as the touchdown PCR, in which we use a higher annealing temperature in the beginning to get an optimal, to get more specific binding of the primers to the target nucleic acid. And initially, when the annealing temperature is a bit high, then only the very specific binding of the primers will occur, and we'll be getting more of the specific target molecules. And then after 35 cycles, the majority will be our desired molecules. And then if you lower the annealing temperature, it's not going to make any difference, but it will increase the efficacy of the multiplication of the products, and we'll be getting more quantity of our desired product. So that is the reason. This is called as a touchdown PCR. Good morning, Abrahami. Yeah, the difference between real-time RT-PCR and the reverse transcriptase PCR. Yeah, this is mainly because of the use of RT. I prefer it. I prefer that the real-time PCR being mentioned as RLRT, RL-PCR better. Real-time PCR is the one which uses probe and which detects the results in the real time. That is, when the sample is progressing, each and every cycle you'll be able to visualize the results. So that is the real time PCR, and this uses the probe. Okay, this uses the probe for measuring the intensity of the PCR reaction, intensity of the fluorescence. Okay, reverse transcriptase, as I told in my presentation before, it is a step. Reverse transcriptase is a step to convert RNA to cDNA. So when you are going to look for the RNA viruses or want to amplify some RNA, first you have to do reverse transcription using a reverse transcriptase enzyme, convert the RNA to cDNA, and then use it for the PCR. Yeah, after boiling, some water will evaporate. The question from Anjali Jaiswal. Yeah. These are the issues which are always there, and we should make sure that we don't overboil things. And the slight difference is not going to affect your results. So just don't worry about the small volume of water that will evaporate after boiling. Shweta, it's not like not you don't have access to touch on PCR. No, it's not at all like that. You can program it in any PCR instrument. You can just call your application support. He will come and help you how to program it on the PCR, whichever PCR you do. He will be able to do it. Like it's just some programming thing. You can do it on any PCR system. Metal nanoparticles. No, don't have an idea about this. Maybe in the future they'll be helpful, but currently the magnetic beads are doing their job. You're welcome. Yeah, the pandemic situation.
Thank you so much, sir. We have many compliments flooded in the chat box. Thank you, sir. Now we have come to the end of the webinar. On behalf of the organizing team, Department of Plant Biology and Plant Biotechnology, myself, Gayatri, propose a word of thanks. Gratitude is one of the least articulate of the emotions, especially when it is deep. I sincerely thank the management, especially the most revered General Secretary and Correspondent, Mr. Manjit Singh Nair, sir, and our beloved Principal, Dr. M.G. Raghunathan, sir, our Vice Principal, Shift 1 and Shift 2, teams of various schools, COE, Media Center, IMS for their constant support and guidance. My grateful thanks to Dr. A.K. Sheriff, Specialist in Microbiology, Department of Medical Microbiology Laboratory, the Royal Hospital, Muscat, Oman, for not only sparing their valuable time for us, sir, but also for enlightening us with about introduction to PCR and its application, discovery, diagnosis, its technique pertaining to it. Thank you so much, sir. It was a witty and very informative session. Thank you for clearing our doubts and enhancing our understanding pertaining to PCR analysis. You have indeed put your best of your effort to make it an event an unforgettable event. Thank you so much, sir. My humble thanks to Surya for his zealous effort, who has devoted a lot of time and energy to make this program a grand success. Next, I would like to thank our torchbearer of this event, HOD, Dr. M. Gopi, for inspiring us and leading and taking us to work forward to make this webinar a grand success. I like to thank our staff members who went an extra mile and shared the webinar's event in all the social media pages. Thank you so much. Our heartfelt thanks to all the participants and students for active participation. With this great message, we move to the end of the today webinar. Thank you. Thank you all. The feedback link is posted in the YouTube description box as well as in the chat box. And the e-certificate for all the participants will send to the respective email addresses provided during the submission of the feedback. Thank you so much. Thank you, sir. Sir, Gopi sir will end the live.